Um, First, I want to thank you, the organizers, for inviting me today to present the work that my lab has been doing for the last 10 years regarding the immune response during Parkinson's disease. During my talk today, we will touch upon the different possibilities that we have to approach immunomodulation as a future therapeutic strategy in Parkinson's disease. First, I want to declare no conflict of interest. When we talk about inflammation in Parkinson's disease, it is well accepted that there exists a microgliosis in the brain of Parkinsonian patients. By microgliosis, we understand the increased number of microglia, which is the macrophage of the brain, or change in morphology, or upregulated expression of different immune proteins in those cells. And that was already shown in 1988 by McGeer, where they presented that an increased expression of HLA-DR, which is MHC class II, in the microglia of the brain of Parkinson patients. Within a few years, several labs, like for example the Tian Hirsch lab, showed that there was increase of pro-inflammatory cytokines in the brain of Parkinsonian patients, and it initiated the concept of a neurotoxicity induced by a chronic inflammation in the brain. Initially, it was thought that the signaling initiating this microgliosis was actually neuronal death. So it was necessary that a neuron die to signal towards the microglia for activation. But during the last 10 years, 15 years, this has changed dramatically. And in the field now we understand that within the brain of Parkinsonian patients, there is microgliosis in multiple areas, not only in those ones where we found neuronal death. So here Imemura in 2003 show that there is elevated expression of MHC class II, not only in substantia nigra or putamen, but also in the hippocampus and in the cortex. And years later, in vivo PET imaging by the group of David Brooks and Nicola Pavese show that the Parkinsonian patients have elevated signal for the ligand of the TSPO, which is a marker of immune activation in the brain, in different areas, not only in basal ganglia, but as well in pons and in frontal region. Not only this, but during the last 10 years, several labs has shown that alpha-synuclein itself is an inflammagen, and it can act as a damage-associated molecular pattern interacting with immune receptors that are found in microglia, monocytes, or other immune cells, and this will initiate a cascade of events that will end up inducing a steroid pro-inflammatory response. These receptors are numerous, as you can see in this image now, but we will focus during my talk rather on the toll-like receptor 4 and 2, which have been extensively shown to be involved in the disease. As I said, the interaction can lead to pro-inflammation, but it can also lead to the degradation of alpha-synuclein. And in that sense, microglia should be a neuroprotective cell because it will avoid, in that way, the spreading of the pathology that has been proposed by the Bragg hypothesis. On the other hand, the degradation of the alpha-synuclein can lead peptides of the alpha-synuclein to the MHC system, and then the innate cells can present this antigen initiated by the alpha-synuclein peptides to T cells, and this can result in autoimmunity, which in fact seems to occur in the patients. Parkinson's disease patients actually have T cells that recognize alpha-synuclein peptides. So my lab, together with several labs in the world, we have been very focused on actually addressing the early response of microglia during alpha-synuclein related neurodegeneration. Using several models, and here I'm presenting you the PFF, the febrile alpha-synuclein PD model, we can show that, for example, in this case, when we inject aggregated alpha-synuclein in the striatum of the rats, we can initiate pathology, alpha-synuclein pathology in distant areas, like, for example, the substantia nigra. This will lead to the neuronal death of the dopaminergic cells, and in parallel, this will also induce an immune response of the microglia that will increase the expression of MHC class II very early, way before the cell death is present. And we can see this microglia will approach the neurons that have alpha-synuclein pathology. So how this happens in the patients, it has been addressed by different studies using PET imaging. And in our collaborators, Eduardo Tolosa and Nicola Pavese, studied a few years ago what happened in the brains of the REM sleep behavior disorder patients. As you know, these patients have a very high risk to develop Parkinson's disease, and they are considered now the prodromal form of Parkinson's disease. So Nicola and Eduardo show that these patients, way before there is any diagnosis of Parkinson's disease, they show an inflammation in the substantia nigra, as shown by the increased binding of the TSPO ligand, and as well a decreased dopaminergic neurotransmission in the putamen, suggesting that the event of neurodegeneration occurs very early prior to diagnosis, obviously, but this is also paralleled by an immune response in the patients. My lab, together with other labs, have been very interested not only in the inflammatory response in the brain, but also in the immune response in the periphery. If we look at the blood immune cells, we can find there monocytes that will respond to alpha-synuclein in the same way that microglia. They will recognize alpha-synuclein, they can clear it, they can get activated, and they can also present alpha-synuclein through the hemiciclase-2 receptor to T cells. This will lead to T cell activation, and in turn, this will lead to B cell activation that will produce autoantibodies. These events have been suggested by studies in PD models that can lead to neurotoxicity, especially the T cell activation seems to increase inflammation and neuronal death, whereas the presence of antibodies seems to be protective because they can enhance the clearance of alpha-synuclein. So there must be a balance in this immune process to achieve neuroprotection. My lab, as I say, are very interested in this peripheral immune response, and we have been studying blood immune cells in different patients' cohorts. We have been interested in the innate cells, and for that, we have been studying the different types of monocytes and the expression of proteins in their surface. The monocytes are normally divided in three groups: the classical monocytes, the intermediates, and the non-classical monocytes, depending on the expression molecules in the surface. And within this group, it's accepted that the classical monocytes are those ones ready to leave blood and infiltrate inflamed tissue in the body, in this case, most likely the brain. So as I say, we studied these immune cells, and we were very interested to see how early the peripheral immune system responds. So we use the blood immune cells from the cohort that I showed you before that Nicola and Eduardo studied with PET imaging. So the REM sleep disorder patients have increased classical monocytes already before PD diagnosis, indicated that there was an increase of those cells that can leave the blood to infiltrate immune tissue. Not only that, but these monocytes have increased expression of proteins related to infiltration and transmigration, for example, CCR2 and CD11B. We also study other receptors expressed by these cells, and we observe that the level of expression of toll-like receptor 4, which I showed you before is one of the interactors sensing alpha-synuclein, was related to inflammation, indicating that the more toll-like receptor that we have in the monocytes, the higher inflammation we find in the patients, and the lower dopaminergic neurotransmission in the putamen. On the opposite side, if we saw more cells expressing CD163, which is a phagocytic marker, we saw that these patients with higher number of phagocytic cells, they have lower immune activation in the brain and better dopaminergic neurotransmission. So indicative of maybe a deleterious role of the toll-like receptor and a protective roles for these cells. Therefore, we show that the immune response involves both the brain, the microglia, and peripheral immune cells, the monocytes in this case, that this immune response was coordinated, so therefore there is a brain-periphery crosstalk, and that it was associated to the neurodegenerative event. But of course, we are interested in how this evolved during the disease, so we also study similarly the monocytes into another cohort of Parkinsonian patients. In this case, what we did was to divide the cohort into early PD, addressing the changes occurring shortly after diagnosis, or late PD. And what we observed was that at early stages of the disease, classical monocytes remains elevated as we saw in the REM sleep behavior disorder. But this was decreased later in the disease, maybe as a sign of exhaustion. These cells were not only increased at early stages, but throughout the disease, the monocytes has increased expression of markers that are helping for the infiltration and recruitment. For example, CCR2 expression. At later stages of the disease, we saw a clear upregulation of the expression of MHC class II, that means the interactive for T cells, and also associated to the diagnosis, we saw an upregulation of the expression of CD163 in the monocytes. So we observed that the changes in the monocytes seems to be dynamic, associated to prodromal early or late stages, and is associated to increased number of cells capable of infiltrate to inflamed tissue, obviously within the brain, and they have an increased phagocytic capacity. But of course, we wonder, are these cells indeed infiltrating the brain? And to study that, we actually took advantage of a specific event that can occur to the CD163 receptor. This receptor is a membrane receptor exclusively expressed in monocytes, but upon an inflammatory signaling, for example, toll-like receptor activation, the receptor is cleaved, forming a soluble form, the soluble CD163, that can be measured in plasma or in CSF, depending where the activation of these macrophages occur. In fact, my lab has shown that human macrophages exposed to aggregated alpha-synuclein become activated and results in the cleavage of the soluble CD163 that can be then measured to be increased in the supernatant. So therefore, we hypothesized if in the patients indeed the monocytes expressing CD163, which are marked to infiltrate the brain, they go to the brain and they encounter inflammation in the brain, this shedding will occur and we will see increased soluble CD163, specifically in the CSF of the patient. And indeed, that's what we found. We found that in early and late PD, we see increasing amounts of soluble CD163 in the CSF of the patients, suggesting that the macrophages that express CD163 infiltrate the brain. There they become activated, and therefore the soluble CD163 is released to the CSF. So was this related in any way to events that are associated to the disease? We believe so, because we actually saw significant correlations of the level of soluble CD163 with accepted markers of neurodegeneration, like, for example, alpha-synuclein or tau or phospho-tau or A-beta-42. Furthermore, if we check at the correlation of soluble CD163 with other markers that are associated with infiltration, we found also that the level of soluble CD163 were significantly correlated to ICAM. ICAM is actually the ligand that binds the integrin CD11B to facilitate endothelial transmigration. And as I showed you before, the monocytes in some patients, they have increased CD11B. On the other hand, we also saw soluble CD163 increase at the same time as CCL2, which is the ligand for the receptor CCR2, also important for chemotaxis, and that was also increased in the monocytes of the patient. So we have an indication here, we believe, that the increase of the soluble CD163 is associated to an environment permissive for the entrance of the monocytes towards the brain. this was actually related to the symptomatic presentation of the disease, because the higher level of soluble CD163 were correlated with worse cognitive capacity in the patients, indicated that macrophage activation actually is deleterious in that way towards the symptomatic presentation of the disease. Therefore, we have indications that these monocytes travel to the brain, but we wanted to study that further. And for that, we actually use the advantage that CD163 itself is only expressed in monocytes and macrophages. It's not expressed in microglia. Within the brain, CD163 is only found in the border-associated macrophages. It is in the meningeal macrophages or perivascular macrophages, so the choroid plexus macrophages, but it's not expressed in the parenchyma of the brain. Therefore, presence of CD163 positive cells in the parenchyma of the brain will be suggestive of an infiltration of peripheral macrophages towards the brain. And in fact, in 2014, the group of gentlemen show that there are CD163 positive cells in the brain of Alzheimer's disease patients and as well of the Parkinson's disease patients, especially in areas like the substantia nigra, locus coeruleus, and dorsal motor nucleus of the vagus nerve, that, as we know, they have a heavy alpha-synuclein pathology. These cells were phagocytic, and these cells has been also found not only in humans, but we can recapitulate that in PD models. For example, Ashley Harms in the West Lab show that in the alpha-synuclein fibril model that I presented you before, where there is microgliosis, there is also a significant infiltration of the macrophages towards the brain in parallel to the presence of CD163 positive cells within the parenchyma of the cell. So indeed showing infiltration of the cells. My own lab has shown that this infiltration also occurs in the toxic model. In the 6-hydroxydopamine model, we saw an increase of the CD163 positive cells in the parenchyma of the striatum. We actually showed that these were truly peripheral immune cells, because what we did was to isolate CD163 cells from naive rats and ex vivo loaded them with a fluorescence dye, and then transfer into the blood by injection into the blood to a rat with a 6-hydroxydopamine lesion. And then we killed the animal 1 day later, and we found that those cells that were injected in the periphery, they actually appear in the striatal parenchyma of the animals. So indeed, we show that peripheral CD163 cells, they actually infiltrate the brain in the PD-like neurodegeneration. So we hypothesized that if we were modifying these cells, we may achieve certain protection in the brain. And to do so, what we did was to design a liposome that was specifically targeted for CD163 by using antibodies against CD163. And for that, we loaded the liposomes with glucocorticoids, with the idea that these liposomes will be specifically uptaken by the CD163 cells, and then these cells will become anti-inflammatory. And when they migrate to the brain, they will be protective towards the neurons. And in fact, that's what we saw. The animals that receive these liposomes targeted for the CD163 have a better survival of the neurons and a better motor performance. So indeed suggesting that if we modify the macrophages that goes to the brain, we can actually protect the neurons. Other labs has approached this idea of the macrophage infiltrating the brain, for example, by genetic deletion of the CCR2 protein, which, as I showed you before, is increased in the patient. If we delete this protein, macrophages cannot enter the brain, and in the PD models, this is actually neuroprotective. But the immune response, as I tell you, in the disease, it's a little bit more complex than we initially thought. It doesn't not only involve microglia, as I show you now, it involves also macrophages. And the MAC class 2 event suggests that T cells are also important within the disease. Indeed, we have shown that in PD models of the disease, CD4 T cells infiltrate the brain. Here I show you the alpha-synuclein viral vector PD model in two scenarios where we achieve pathology in the terminals of the striatum but no neuronal death, or with pathology and neuronal death. And as you can see, microgliosis occur in both of the situations without cell death or with cell death. And this peak of microgliosis coincide with the entrance of CD4 in the brain, suggesting that there was a direct interaction between the microglia and the T cells infiltrating the brain. So this actually is supported by findings in the human with Parkinson's disease, where we observe that there are T cells that, as I said before, can recognize peptides of alpha-synuclein and respond accordingly. This has been shown by the Sulzer and the Sede lab. The T cells actually can respond with different effector capacity. There are the Th1 and Th70 are considered normally pro-inflammatory effectors, whereas the Th2 and T regulatory cell are anti-inflammatory and therefore associated to a neuroprotection. Interestingly, in human patients of Parkinson's disease, we found that there is an increase of the Th1 and Th17, which is the inflammatory form of the effector T cells. My lab actually approached a neuroprotective strategy aiming to enhance this T regulatory population that can lead the microglia to a more neuroprotective status. To that, what we did was to design an alpha-synuclein vaccination that resulted in the expansion of the T regulatory cells with migratory capacity. So we approached this vaccination in the alpha-synuclein viral vector model, where alpha-synuclein overexpression leads to pathology of the terminals in the striatum. And as you can see, the vaccinated animals show a significant less pathology in the striatum. And this was not associated to a lack of microgliosis, because there was microgliosis in these animals. But what we observed, it was that this microgliosis was specifically associated with an entrance of T regulatory cells in the brain, suggesting that the entrance of the regulatory cells will help these microglia to become neuroprotective and help the survival of the neurons and the decrease of the pathology. Indeed, signs of neuroprotections were confirmed by the increased expression of GDNF, that, as you know, is a growth factor that the microglia can release to help the neurons to survive. This was only present when the vaccination approach was used. As well, we saw a significant increase in filtration of IgG autoantibodies against alpha-synuclein, and this has been suggested to, as I said before, increase the clearance of alpha-synuclein and be neuroprotective. In fact, as you know, there are several companies addressing the capacity of antibodies to be neuroprotectors, and they are nowadays in phase 1 trial. So indeed, the modification of the T cell induced neuroprotection. Other labs have approached similarly to us T cells. The Harms lab has been very focused in avoiding T cell toxicity, decreasing effector Th1 and Th17. Like, for example, recently they did by using this drug, fingolimod, that is approved for MS, to decrease the T cell response in the model, and they actually saw neuroprotection. The Gendelman lab, on the other hand, was like us, approaching to enhance tolerance, and they have used different strategies in the models to increase T regulatories and have shown that it's neuroprotective. And they are nowadays participating in a phase 1 trial using GM-CSF agonists, also with the aim of increased T regulatory cells. So I have showed you today that the immune response in PD occurs very early, prior to diagnosis. This immune response involves brain and peripheral immune cells, is coordinated and associated with neurodegeneration. The changes in monocytes is dynamics during the disease, And when the monocytes are marked for infiltration, the brain then seems to participate in the immune response and influence the neuronal health. So if we modulate, we achieve neuroprotection. And also the MHC expression leads to the interaction of T cells that also infiltrate the brain, partaking into the inflammatory response. And therefore, if modulated, we can also achieve neuronal protection. So to end up today, I show you this slide that I know is very busy, but I just want to show you that the immune response in the disease is rather complex, much more complex than we initially thought. There's not only microglia, but also macrophages and T cells and other cells that I have not talked today, like NK cells and neutrophils. The advantage that we have in the PD field is that other diseases with immune component have already tried several strategies to address these different roads that I present you here. So we could take that as an advantage and try to use those drugs that have been already approved for example, Crohn's disease or MS or psoriasis. In fact, there are several drugs that, as I say, they are in PD trial, like the enhanced Tregs and the vaccination, both passive and active. So with that, I'm going to finish. I want to thank especially my lab for the work that I presented today, and my collaborators essential for this work, and finally to the foundations that support my research throughout the year. And thank you for listening.